Hi, this is Miss Litton, and this is my wonderful APBio Period 1 class. Say hi.
Hi.
And they have leaves sitting in front of them that they're getting ready to paint in order so we can count the stomata on the underside of leaves. I want to back up our presentation just a little bit to review. We are under Chapter 25, Flowering Plants, Nutrition and Transport, and we are in Section 25.3, Transport Mechanisms in Plants. And so the two vessels that we're looking at are xylem and what? Phloem. Okay? And which one is dead usually at maturity?
Xylem.
Xylem is usually dead at maturity. Okay? And so when we look at xylem, the biggest, widest pipes of xylem are called vessels. And it looks just like PVC pipes stacked on top of each other. And then tracheids are the thinner ones here on the side. And we'll talk about the mechanisms about the way water moves through those in just a moment. Here, you can see actual xylem. These are tracheids over here on the side. Phloem has to be alive at maturity. And we'll talk about the pressure flow hypothesis. You want to remember structurally you have sieve tubes, and then you have a companion cell that is attached to it. It's only the companion cell that has the nucleus. This sieve tube does not. And it is hypothesized that the companion cell controls the sieve tube member. And did I ask you this question last time? I'm going to ask you now. For some of you, this may be the first time you've used fingernail polish. How exciting. Josh, Maria, Benjamin. What's going on? Oh, that's scary. All right. So most of you got that. Xylem does not include companion cells. Where do you see companion cells? With what? Phloem, which I just went over 30 seconds ago. So check your BioBuddies clicker right now. Do they get some chastisement or do they get some praisement? All right. So now we're focusing on specifically water transport. Where is the source of the water coming from? From the roots, from the soil specifically. Yeah, from the soil. So we got to get water from the soil into the roots. And what am I, oh, it's labeled. What is this right here? A root, what? Hair. Okay. So when you talk about water movement, you're always going to relate to water potential. Pure distilled water has a water potential of what? Zero. And water potential is a factor of what two items? Solute potential and then pressure potential. Okay. And you know the equation for solute potential. Solute potential equals negative ICRT. Go, Ruthie. Okay? So you could calculate that. You've done a lab on that in calculating water potential, yes? So we're going to hope then, if you want water to go into your plant, it has to have a higher water potential where? The soil or the plant? The soil. So it'll go from a higher water potential in the soil to a lower water potential in the plant. What happens if, like, we don't experience this, but apparently other places in the United States have weather. And apparently it snows, and this is what's like frozen rain that falls from the sky, and apparently it piles up in places. And it can make roads very, very dangerous when you're driving on them, especially black ice. And what they'll do to get rid of the snow is they will sprinkle salt along the snow and plow it over to the side, and it causes it to melt. And what could that do to the plants that are surrounded with salt water? What could that do? Make go?
You could dehydrate the plants because now the water is central outside of the plants. It's hypertonic.
So what's happening to the water potential outside the plant? It's going down, and because of all that salt, making it very hypertonic, so water would leave. Okay, so could you ever use too much, if you were like planting a new garden in your backyard and you were using the fertilizer that they gave you, and you're like, oh, well, they say to use like a cup of fertilizer for this one plant. I'm going to use 10 cups because I really want my plant to grow. Could you ever use too much fertilizer? Yes. Okay? Too many things in that soil, you can make it very hypertonic and water would leave your plant. Okay? So that's the basic part to get the party started. So water is going to move in, and the soil, we know, remember we learned about the proton pump already. So by pumping protons out, right, you can use that to escort, co-transport other molecules in to concentrate minerals inside the plant in order to lower the water potential so water flows in. Yes?
And plants that don't necessarily need dirt, like green stuff?
Yes. So they have to have enough of the nutrients in the water that they're in. Remember we looked a little bit at hydroponics? So there must be something dissolved in the water that gets them everything that they need. Okay. So when water moves in, and it moves into the steel, into the xylem, it'll start, the only place it has to go if it can't move out, remember we already had the discussion about the Casparian strips, yeah? And the only place it has to go is what? Up. And when it goes up, you can see evidence of this, it's called guttation, and water is literally getting squeezed and pushed out the stomata themselves and dripping out of the ends of the leaves. And that's evidence of what's called root pressure as water is flowing into the roots. So on your notes, we have water transport, root pressure, water moving upwards as a result of following the minerals into the root. As a result of following the minerals into the root. Root pressure forces water up and out the ends of leaves. It's called guttation. It's called guttation. Now, we have already talked about the TATC theory, or another name for this is the cohesion tension model of what motivates water to move. So we talked about one of the T's is T for okay. Okay, the T is for transpiration. Could Blue please explain to Slate what is transpiration? Give him a definition of that.
Wait, so it's literally how the soil, so it goes into the roots and there's so much water.
Weird.
To the water.
Okay. All right, so transpiration is the evaporation of water out open what? Stomata. And we know that one water molecule is always connected to another water molecule via what kind of bonds? Hydrogen bonds. And the reason why water has hydrogen bonds, forms them easy, is because what kind of molecule is it? Starts with a P, polar. And the oxygens are always slightly negative because they have a stronger electronegativity and they pull on hydrogens, electrons, and keep them a little closer to them. So the oxygen is electronegative, the hydrogen, is slightly negative, the hydrogens are slightly positive. So that attraction then, because it's a polar molecule, that weak attraction, that's a hydrogen bond. And when a bunch of water molecules are hooked together, what's the word we're looking for? Cohesion, okay? And that, so when one water molecule evaporates, it pulls on the next water molecule, which pulls on the next water molecule. Well, this would be a really long chain of water molecules, and you would tend to have breaks and gaps in it. But what kind of holds all the water molecules up is that the xylem is a polar surface, the xylem walls, and so it can attach to it, and when it attaches, the water attaches to the side, that's called what? Adhesion. Okay, so that gives us support. And then this pulling process, just like sucking on a straw that's in a Frosty, that pulling process, that's tension. going from that higher water potential to a lower water potential. It just keeps getting pulled up and up and up. So that's why it's called the cohesion tension model, or the TATC. So go on to your notes. Cohesion tension model of xylem transport requires no energy by the plant. Requires no energy by the plant. There's a higher water potential in soil than in the roots. Transpiration is the evaporation of water out open stomata. Evaporation of water out open stomata. Cohesion is water molecules are all connected through hydrogen bonds. So as one evaporates, it pulls on the next, etc. Adhesion, water column is supported by adhering to xylem walls. Not it. Whoever's not it, you go first, take turns explaining all 4 words. So one of you takes one of the words, one of you takes another, and just work your way through. Go ahead.
Transpiration, which is where the water like about and then adhesion to the water. I'm sorry.
Could you write an essay on that? Good. That would be a good essay to write it on. Did I share with you potential essays? Did you see that massive document I spent many hours working on? Okay. All right. So a key part of all this, did the xylem cells, I'm not talking about the root cells, but did the xylem cells need to be alive to make this happen? No, in fact, they kind of get in the way if they are. If you just have an open pipe, transpiration will happen. Water will evaporate. So a key part to this is whether or not transpiration can happen. In order for transpiration to happen, the stomata have to be what? Open. And why are the stomata open? The plants need CO2. They're generating a lot of oxygen, right? But they need CO2 for gas exchange. So you have to balance the need for CO2 with the fear of water what? Loss. And that's why you have sunken stomata, or stomata with hairs on them, or you have them on the underside of the leaf, or fewer stomata. Those are all variations that you can do. Here you can see real stomata, or stoma, and then you can see an open and a closed. And you know what? We're going to use a very similar strategy to open and close them as we did to move any kind of mineral. So we'll talk about opening. I didn't give you the fun facts. Here we go. Fun facts. How much of water do you think is taken up by roots and lost due to transpiration? 90%. 90%. And transpiration has a huge impact on climate. 50 to 75% of the rain in the Amazon originated in a plant. What are we doing to the plants in the Amazon?
Killing them.
Yeah, we're killing them. So smart. Okay, and then number 3, cooling. When transpiration takes place, it's cooling the plant and the surrounding area. Just a few fun facts for your day. You can share those with your friends at nutrition. All right, so opening and closing of stomata. Guard cells create space of stomata. Okay, I made this red. I made it red every time it was in these notes. I made it for proton pump. So we saw the proton pump before for mineral uptake in the roots. Now we're going to see it proton pump again. And I'll let Slate go ahead, and I wrote it all out for you. Take care of B.
So,
it's in your notes right there. All right. So in this case, we're using the proton pump not for co-transport to escort something in. We're using it for exchange. So you pump hydrogen out, so a positive ion goes out, so another positive ion now wants to go back in. And when the potassium ion comes back in, who follows it? Water. Water follows it. Water moves into a storage area inside that cell. What is that storage area called? Vacuole. Vacuole swells, pushes against the cell walls. Either in the two guard cells are joined together. That swelling causes the stoma to open. And when it opens, you allow for gas exchange. You also allow for water to escape. And the reverse of that closes it. The reverse of that closes it. Yes?
Can the vacuole itself burst if there's too much water?
What will prevent it? Cell wall. And what is that called? Turgor pressure. Boom. All right. So C, you do the reverse to close it. And then I gave you some factors that are influencing the process. The amount of light, the presence of CO2, a hormone we're going to learn about later, abscisic acid, the internal biological clock. They will look at stomata, and depending on time, it's like you can tell the time by whether they open or close. They will open and close regardless of light. They'll open and close regularly, even in the dark. Okay, that's why this, though plants are not always everybody's favorite chapter, but you can integrate so many different concepts into just talking about water transport. Okay, so once we learn about hormones, we'll revisit this again. Okay. And internal clocks. And temperature and humidity and stress. By stress, it's not like the plant has a test to study for, it's water stress. No, on whether it's opening or closing. I know, that was super, super funny. All right, now, this in essence, it's just in a little experiment to show that if you had a little mercury at the bottom and water on top, that you could move the mercury up against gravity and you could measure how far it moves up. And from that, you would know how much water had transpired. You could change it, if you had two different substances, you could measure the amount of milliliters. All right, now here, let's start talking about phloem. So, organic nutrient transport, role of phloem. Phloem can move in any direction. Now, why am I saying that? What must that mean if I say phloem can move in any direction? Xylem must not, right? Xylem works in how many directions? And what is that way? Up. It only flows up. Now, let me give you a little tip, though. The water can flow up through the xylem. It can get recycled and go back down through the phloem. Okay? But phloem moves wherever it is needed. It moves from the source to the sink. Now, the source could be the leaves. Look at our tree we looked at the beginning of the school year. Did it look different then? Yes. In our quote-unquote winter, 40, okay? And the leaves have all fallen. So the leaves are not a source of sugar for that plant anymore. Where is the sugar? Stored in the roots, underground. Okay, so now the source for sugars is in the roots for that plant, and the sink is wherever this plant needs sugar in order to keep alive. Some of the branches may die out there on the end and fall off, but whatever it's keeping alive, it's got to go from the source to the sink. When we started school in the fall, it was all the pretty leaves on there, and the source would be the leaves, and the sink would be down in the roots. Or growing branches would be a sink. As it grows up, it's going to be consuming those sugars. And basically what happens is you take your sugars, you pump them into a neighboring cell. This is why you need to have it be alive, into a sieve tube. You make the neighboring cell very hypertonic. Who's going to want to follow it? Water. So water follows, increasing the turgor pressure, which pushes it into the next one, which pushes it into the next one, which pushes it into the next one. Because you keep pumping sugars out of your source, and water keeps following it. And that's why it's called the pressure flow hypothesis. Okay, it is push powered, whereas transpiration is what? Pull powered. Do you see the difference? Okay. So on your notes, here you can see they've traced sugars moving through a leaf. here an aphid has tapped in to the phloem so he can have a little snack, and the pressure is flowing so greatly that it's flowing right out his cloaca. That's sugar going in his system and getting squirted out the other side due to that pressure. And what they like to do is they allow aphids to go into the phloem, then they break off the bulk of the aphid and just leave it stylet that's tapped into the phloem, and they can measure how much fluid comes out to see how much fluid is moving in the phloem. So using a biological organism in order to test that. If you like maple syrup, they wait until the sugars start running in the winter, and that's when the sugar's coming up from the roots. That's the sweetest sugars to get. They tap in and drain off the phloem in that way. So here you can see the process. I put it out 1, 2, 3, ping pong back and forth. Blue will go first with a 1. Go ahead. Yes, I do. Yeah, you should make it thin. You guys, you made your fingernail polish thin, right? Because if you make it a big bubble, you are not going to be able to see through it. It's okay, you never used fingernail polish before. You would. No, you're just putting your top coat on there. All right. Should have said that. Okay, so continuing on, go to can move in any direction. The source is where sugars originate. Could be the roots if it was stored, or leaves producing sugars. The sink is where sugars are being used, like a growing shoot or stored roots. It contains mostly water and sucrose, but can carry amino acids, hormones, RNA, and proteins involved in defense and reproduction. The pressure flow model of phloem transport. What do you see first? Proton pump again. Hydrogen ions pumped into mesophyll cells. Hydrogen ions pumped into mesophyll cells. The hydrogen ions then escort, it's +1. You know what I mean by that? On invitations, you're +1. Okay, it's +1. Sucrose, it's not a biological term. Hydrogen ions then escort sucrose into the sieve tube cell, and that is called what? Cotransport. Water follows the sucrose into the hypertonic sieve cell, and then pressure builds. Flow of sucrose moves from high pressure source to low pressure sink. At the sink, water is reclaimed by the xylem. At the sink, water is reclaimed by the xylem. Tape up from the back. Use the tape. Maria? Okay. Oh, whoops. Okay, guys, we better talk about this. It's kind of huge. What main force drives absorption of water, creates tension, and draws water through the plant? You're right, it is transpiration. You can have cohesion in a lake, right? That's not going to pull the water up. Your source is the sun, right? The low water potential out in the air, and it's pulling that water up. That is key. That one is shock and awe for me. Okay, that you're not getting that. So you want to make sure you understand transpiration is the key thing that's pulling that up. the force that drives. It's not cohesion isn't a force. Cohesion is just one water molecule connected to another water molecule. What's powering all of this is transpiration. Okay, check with your bio buddies, see if they got that right. Did I give you summaries of this whole chapter? Okay, and what you have to know? Okay, much better on this one. Good job. Sugars are active. Water always flows by osmosis. Water always. What is the definition of osmosis? Diffusion of water across a semi-permeable. Okay, just checking. Okay. So that is the end of this chapter. I'm just let me put a stop to this. Hope you're having a great day. Have some toast.